SmartLab University lecture series, but also even more to host Rick and to introduce him, because I have been following him in the last, I don't know, 3-4 years, and always being inspired how he just takes a device and brings it to the so-called now PyLabRobot, let's say open source infrastructure. And just to give you a quick introduction about Rick, I hope he also would mention a couple of things. He is at the moment is working as a programmer at Retro Bioscience, and I would just really say big thank to Retro Bioscience that it's even allowing him to continue working on this amazing open source project, PyLabRobot. And at the moment, you might, if you have not been seeing the work of Rick, hopefully you should get interested now what he is doing and why it's so important. But please follow him on LinkedIn, or there is really also amazing YouTube interview with him. I would share it in the channel to understand also why he got excited on this and how his problem solving, how to say, interest has never given him a peace when he saw the problem of what scientists facing when they lack full control onto the devices we have into the lab. And I think what is really exciting to see how all the work which Rick is doing with also bringing a lot of people on board on this open source community, how now even vendors start realizing, okay, we should do something, we should really help and get away of this closed mindset of locking scientists in the vendor softwares and so on. And I'm really looking forward to the talk. Rick, maybe you can also share your presentation so we can get started. Really looking forward to your talk. And I guess we can also keep it as an interactive if you don't mind, or please tell us what do you prefer. If you want to keep the questions at the end, or we can also have it a bit more interactive.
Maybe questions at the end sounds good.
Okay, we do it. We keep the questions at the end. You just need to find the right screen.
Yeah, I think here we go.
Okay, then Rick.
Can you see it at full screen?
It's not yet. Just a second. Can you try again?
Share screen. This should work.
Yeah, perfect. It's just maybe a second to load the high resolution version, but it's coming. We see it. Perfect.
All right. Yeah. Thank you, Nikolay, for the introduction. Herzlich willkommen zu meiner Präsentation über PyLabRobot. I guess for the Germans. I hope my programming skills are a little bit better than my German skills, so I'll just talk about that for the talk. Before I start, I'd like to acknowledge that even though I'm the one presenting this work here today, it's not just my work, it's actually done by now 50 contributors to the open source project. And as Nikolay mentioned, we have some fantastic industry supporters like vendor companies, distributors, and different people using it. So I'd just like to acknowledge, and also that together with Camilla, we are kind of leading and maintaining this project. So specifically today, I'll be talking about, first of all, what actually is PyLabRobot, what am I actually talking about? Then give a very brief history, talk about some applications, then go into briefly how it works internally, talk about why I am such a big fan of open source, and then kind of shed some light on the future plans. And please ask questions at the end. So as an example, what is PyLabRobot? I just like to kind of describe it as like it's a Python SDK for lab automation. And SDK means it's a software development kit. So what does that actually mean? Maybe it's a little bit vague. So just here's one example of a PyLabRobot code snippet. It's just like a Python function here. It's not important that you look at the specific syntax. I just kind of like to show the actual thing that I'm talking about so people can understand in like non-vague terms. So PyLabRobot is just kind of a library where people can program their lab automation machines. So in this case, I'm defining a transfer function where I'm just taking some liquid, for example, from a source to a target, which is just like two calls like aspirates and dispense. And then if you want to, for example, do a serial dilution, which is just a repeated series of transfers from a source to a target, looping over multiple tubes, you can just define that very simply in a loop as follows. The specific syntax, again, is not important. I just want to show some example of the stuff that I'm talking about. We can also work with multiple machines. So let's say, as an example, you want to move a plate from the liquid handling deck to a fridge. You can say like LH pick up resource and then drop the resource to drop it somewhere else. And then you can, for example, wait for the fridge to recognize the plate. And then once it is recognized on the transfer station, you can take it into a specific site, for example, the smallest one that's available. I would like to talk about a brief history. And to me as a programmer, it always seems like, isn't it obvious that you want to have a kind of an API like this? Well, the answer is no. In a lot of cases, the machines, when you get them, they come with this very complicated vendor software that has like a billion options. And sometimes I like to describe it as it's more complex than like the cockpit of an airplane in terms of the number of options and stuff that's available. And also importantly, every machine that you buy typically actually comes with a computer because the software is so difficult to install that they just rather give you a computer, and it's also very, very specific to the machine that you're using. And so if you describe this kind of abstractly, it's for scientists to use automation, they kind of always have to talk to an automation specialist, which is a specially trained person, because automation is so difficult and inaccessible. And then automation specialists, they interface with different kinds of interfaces. So typically people are trained for either, like, for example, Venus or EvoWare or some of the other software. And there are many downsides to this that I kind of listed here. So the iteration time is long. The software is expensive. You must use Windows. You need to buy a robot to even get access to the computer with the software on it. And so what my lab did, the Esvelt lab in the media lab, is this project is started by Dana. He created a Python interface to Venus, which is called PyHamilton, which you've probably heard of. And so even though Venus is still closed source in Windows only, they actually did kind of this very nice hack where they were able to use Python to interface with Venus, and then use the robot through Python. So at this point, you can actually program them like a programmer would, and get access to source control and all these different things. But under the hood, I was thinking, Venus is just sending some commands over a wire to the robot to make it do things. And physically, if you think about it, it's connected by a USB cable. So why is there, what is the reason that I would not be able to send those commands myself instead of having Venus send them? Why could I not send them myself? And so that is kind of the core idea behind PyLabRobot, is that for every machine, we can just send the commands that the machine understands directly from PyLabRobot. So it is completely independent of the vendor software. You don't need to have Venus installed. You don't need to have EvoWare installed. You don't need to have ViewWorks installed. And just PyLabRobot is enough to talk to the machines. And then there's one library, sort of going from the right to the left here. That is PyLabRobot that talks to all these machines. And actually, I put the unlocked icon here because it's open source. So you can just go in at every layer of the stack and edit code and use it and modify it as you wish. And it works on every operating system because it is just Python code. We're not reliant on any vendor software to run. You just send like USB commands. And so PyLabRobot, it can be used directly, but also it is kind of made for other tools to use PyLabRobot as a foundation. So as an example, you can have AI assistants, which I'll show a little bit later, use PyLabRobot. You can get protocols that are agnostic to robots and just run them. You can use it interactively, as I mentioned, and then you can also use third-party tools to talk to PyLabRobot. So you don't have to use it directly. You can use it kind of as a foundation. And what this allows then on the left side is that you no longer need specialized people to use automation, because these tools like AI assistants are very accessible to everyone. So that means that now scientists and programmers and engineers and students, anyone can just kind of benefit from automation. And also the labs, they're now connected. Whereas if you see on the top right here, in the previous case, it was very difficult to share methods between like a Hamilton and a TCAN robot. But in this case, because we have a universal layer over the machines, we can actually share different things between different labs. So why is this actually useful? Maybe it just sounds like some theory. So I'd like to show some examples. So I'd like to first of all preface this by saying I cannot really show like people-specific protocols because they're often proprietary. We can oftentimes collaborate on the infrastructure, but the specific applications that people actually do in a lab, they are oftentimes proprietary, and I actually prefer to not know about what people are using it for. So I just show some examples that are kind of toy examples, and then I think you can use your biological intuition, which is probably better than mine, to think about how this can be applied to actual useful lab things in biology. So first of all, I have this thing that I call the PyLabRobot Art Studio, where this was created in the Media Lab, where you can use this online editor where you have access to different colors and kind of make drawings on this microplate. So this is just like a 12 by 8 microplate, and you can use different colors to paint something here. And then what we can actually do is have an OpenTrans paint this on a piece of paper that we put on a microplate. So using the different color dyes here. And you can use some of the biological principles here. For example, you do not want to use the same tip for different colors of paint, because that is kind of analogous to cross-contamination. So you prefer to pick up a new tip every time you start a new color. However, as long as you're doing the same color, you can actually keep using the same tip, and that is just more efficient. And what is interesting about this to me is that at the time that I was writing this editor, there's like a billion different drawings that people can make. And as a programmer, I had no idea how people would actually use it. Like, what would they draw in this program? And still, I was able to write this program and make it work. So that kind of shows that you can have protocols that are non-deterministic. So for me as a programmer, I had no idea what was going to happen. And so you can write a program to allow people to do these things. Another thing that we did at Retro is you oftentimes work with sealed plates, and manufacturers like to sell you their $20,000 desealer for a plate, which just takes the wrap off. But sometimes you only want to use a plate once, which means that just actually piercing the foil is enough. And so we were able to develop this this kind of step called pierce foil, where we take some pipetting channels and actually just pierce the foil. And then we use 2 channels on the side, if you can see in the animation here, to hold the plate down, so that as you move the channels that pierce the foil up, they will actually come up instead of dragging the plate with them. So the hold down channels, they're very important. And this is allowed in PyLabRobot because we can have very like low-level control over the machines. We can individually, for the channels, instruct how they should move. And that is what enables this kind of interesting behavior that means we don't have to buy a desealer for this particular application. We can also 3D print custom labware. So again, because we just tell the robot to like go do operations at X, Y, and Z, we can have full control over the labware model on the PyLabRobot side. And in this case, we 3D printed this tilt module, which in total, I think I'm allowed to say costs around $15, which is much cheaper than a $10,000 OEM part for tilting. And this is just actuated by a pipetting channel coming to push the plate either into the tilted position or into the non-tilted position. We can also be better than OEM software in other ways. So for example, with a citation, to image a plate, to image 72 walls in a plate, it actually takes over an hour to take these images. And by going to the low-level commands of these machines, I was able to spot some inefficiencies and actually optimize those and then deploy them to the users. So on the first day, I found a lot of inefficiencies and got the time down to 25 minutes. And then the next day, I was like, I can find some more inefficiencies and shaved another 8 minutes off the imaging time. And then because it is open source, I can just push this to our repository and people can start using it as soon as I find and publish these things. And in this case, it's actually very important to be fast for our application because the incubation time for this particular protocol is actually 1 hour. So if it takes you another hour to image, that means that by the end of imaging, like your 70th well is going to look biologically different than the first well, which means that you can actually just not do this if your imaging takes an hour, because the biological data between the first and last sample, they are incomparable. And so in this case, we're not just being faster than OEM software, we're actually doing things that with the OEM software would not be possible. With OEM software, we could just not image the plate as a whole plate, whereas with PLR we can. Another example that is probably my favorite, which is made by Camillo and therefore called Z probing, because he lives in the UK, is this tool where we can use the CLD function of a star to probe the surface of different things in the deck. So there's this video where we can just repeatedly probe the stuff on the deck, and you can see it being plotted here, which actually turns the star into a CMM, like a coordinate measurement machine, which I think is very cool. And actually, when we showed this to people at Hamilton, they were like, we thought we were just making liquid handling robots, but apparently we're also making CMMs. So that the stuff that even the manufacturer did not imagine that you could do with their machines. And again, this is enabled by the low level control that we have over the machines. I'd also like to show the visualizer. So for PyLabRobot, we have a model of obviously everything on the deck and like where different things are. And the visualizer just shows you, like for example, for the carriers, where they are, for the plates, which are on the left here. The color intensity is proportional to how much liquid is in a well, so that you can visually see kind of where PyLabRobot thinks the liquid is. And then on the right-hand side is the tip rack, so you can see green is where tip is, white is where no tip is. And this can be very useful when you're developing protocols away from the robot. So for example, at home, you can develop these protocols and kind of see how it would work on a robot just by inspecting it. But also you can use this when you're actually doing a protocol in the lab and you want to see, does PyLabRobot's model of what is going on actually match what I see on the robot in front of me? And also this can be used to, for example, show users, like, this is where PyLabRobot expects your tips to be, and make sure that when you load the robot, this is where all the tips actually are. And this is another very cool example that we did recently in collaboration with the Biomni team at Stanford, is actually agents writing PyLabRobot code. So the Biomni team, they make this AI agent called Biomni, which is a general purpose biomedical AI agent. So essentially what that means is it's kind of like ChatGPT, but as you know, ChatGPT has like tools, for example, to make images and search the web and execute some code. Biomni is like that, but it's specifically tailored towards biology, where it is not just those tools, but like hundreds of other tools. Like it can search data sets, it can analyze data, it can use like genome viewers and all of these different tools. And then we worked with them to actually give it hands, so to speak, to make it able to write PyLabRobot code and test this in silico before it gives the code to the user. So it can actually iterate on these things before giving the code to the user. And as a result, when it responds to your query, the code has been validated by Biomni on the backend, and it can run perfectly on an actual robot. So you can see an example here of just a very simple protocol moving water from wells like A1 through A3 to B1 through B3. And what is also cool about this to me is that these tools, Biomni kind of relies on the large language models, and PyLabRobot is a separate library. So as we make those respective tools better, the whole system is only going to get better. As an example, the AI models are going to get smarter. And so Biomni will get better at writing more complex protocols. And also, as we give more functions in PyLabRobot, for example, we add new machines, Biomni will automatically be able to use those tools in the future. Finally, I'd like to just kind of zoom out and talk about why I think Python specifically is a good language for interfacing with all of these things. So first of all, Python has a very big ecosystem of existing tools. So for example, if you want to integrate with Slack or NumPy or Pandas or any of the analysis tools, everything is kind of available in Python. This, like the entire modern AI ecosystem is built on top of it. Also, you can get full control over the machines, and this is mostly because we are able to send the raw firmware commands to every machine. and just it's like open source the entire way, so you can just go into the code and edit any part of the function. You're not reliant on just black box vendor stuff. Control over your protocols, and that is because the text files are just nice to read, and you can use Git and all these different tools. Also, Python is free to use. Like you can use any computer. When I give someone a Python protocol, all they need is Python, which they can just very easily get. Whereas if you give someone like a protocol in a specific language, you might imagine it's very difficult to get like some piece of vendor software sometimes. Also, just code allows you to build abstractions and modules. You can have APIs that are agnostic to different machines. For example, using like Lapper on an Opentrons to on a Hamilton. And very important, ChatGPT and other LLMs can help write your code, where they really are fluent in Python, but not fluent in Venus, because there's just so much more examples for it to learn how to write Python code. And then finally, and most importantly, it is easy to build higher level software with Python, which I'll get into a little bit later. So I've hopefully convinced that this is something that is useful and worthwhile. So maybe now I'll talk about internally, how does it work, just very briefly. On the highest level, we have kind of 3 layers, which are the interface layer, an execution layer, and then the physical hardware. So the interface layer is the hardware agnostic part, where we kind of realize for every machine that there's very similar functionalities across every machine in this class. So for example, for a liquid handling robot, there are only 4 operations, which are picking up and dropping tips, and then aspirating and dispensing. And as long as you can do all 4 of those, you're qualified as a liquid handler. And so on the interface layer, we can just have those 4 functions, and then have like nice user APIs, and just have the users write code that targets liquid handler using those functions. And then when it's time to run those protocols, we can choose a backend, as we call it, an execution layer that will actually go and execute those specific operations. So in the case of this highlighted here is, for example, using an evo, we use the evo backend, where the liquid handler is going to send the aspiration command, which is abstract, to the evo backend, and then the evo backend will send a firmware command for aspirating to the evo. So in this case, it's like C5, TMZ, different bytes essentially that are sent to the machine. And then we can do the same thing for plate readers, as is shown here, where the operation is just read a plate, like absorbance at a specific wavelength, which is then converted to a machine-specific command. We can do this for skills. We can do this for incubators. We can do this for robotic arms. Kind of any machine that follows a similar pattern. We also have a big library of LabVAR definitions. So this we call the PyLabRobot resource library, which is also you can see on our website. And it's important to highlight here that these are actually crowdsourced from different people, just using the LabVAR and looking at technical drawings. And we really put a lot of effort into making sure that the LabVAR is very accurate, which is a little bit of a different approach than I see people sometimes take, where for us, calibrating the LabVAR, sometimes it takes like half an hour to get like all the specific dimensions correct from the technical drawing, or sometimes even manually calibrating the LabVAR. And the reason for that is that if you don't do this, you end up kind of with what Camilla describes as a house of cards, where if you imagine a plate and then you use it in one protocol, but then in the second protocol, you think, oh, I have this plate that is kind of, very close to what I'm using. So I'll just copy it here and name it like, PCR plate V25 December version 2.0. And then I would just use some offsets to make sure that the pipettes go to the correct location. Well, in that case, if you move the plate to a different part in your protocol, for example, you use the plate on liquid handler and then you move it to a plate reader, suddenly you need to redo your whole offsets thing. And so everything kind of falls apart. Whereas if you have very, very accurate LabVAR definitions, you can actually just take it. And the physical truth of the plate is the same everywhere. And so if you base everything off that, you don't really have to use offsets. And this is particularly important with systems like the Biomni's AI agent that I showed earlier, because it can iterate kind of on the computer side, but on the physical side, it does not know if the channels are going to the correct place. And so we have to make sure that when a model specifies I'm using this particular plate, that we can actually know where to go on this plate. Again, because the models, they cannot really know what your custom offsets should be. But in reality, if you think about it, if you know what the deck looks like, if you know what the carrier looks like, if you know what the plates and the walls look like, you can just sum all of those things, and you should be able to arrive at good conclusions, which is what actually we're able to do. So then, why should you use and contribute to open source software? Why am I not selling this to people instead of just giving it away for free? So you can say there's a philanthropic argument, but also we live in like a capitalist society. So let's just use a correct assumption that you're greedy. So first of all, by making your integrations open source, they will be kind of maintained for free, because as other people start using them, they get an interest in this working well, and so they're going to use it and test it and contribute it back to you. And also, your integrations will be used by many people in many different labs, and so they will become more robust. So as an example, when we have different interfaces to machines, and the machine is like a slightly different configuration for someone else, that you'll not be able to find if you're just using it in your own lab, but then other people are able to find them and make them more robust. And this is very important in lab automation, because we know that every error is very expensive. And so even if you can share the amount of errors by many people, it'll become very cheap for you. If every error occurs like once in 1,000 times, you can wait for this to happen to you after 1,000 times of trying, or you can kind of spread this across different people. Also, it's important that the user can actually fix things instead of having to call a phone number. So this is a very short iteration loop, like when there's a bug or something happening, you can just literally go into the code and see what's going on. Although we are also pretty fast, so if people report bugs in the forum, we can usually also fix those pretty fast. And then most importantly is when you use open source software, you're already be building on this particular program, whereas other people will have to catch up if they decide to switch later. So as one example of this, let's say that we keep, for example, the citation driver just internally. At one point, someone is going to realize that they should open source their citation driver. And then other people are going to contribute to it, and it's going to become better. And then we are kind of stuck with this driver that is worse than the open source one. And so we'll have technical debt, and we will have to switch and catch up to get the better results. And so as a result, you'd rather be the person leading this and showing this driver to the world, so that other people actually contribute to it. And you don't have to catch up because you're already using the best version that exists. So what is next? I think Python APIs, they're very interesting to me, but I'm not unrealistic. I think they're probably not the best interface for wet lab biologists. And so as a result, we need to build better interfaces. And I think this is going to be the work of many people to build those better interfaces. And so how do we get there? I think we get there by just building very good tools for developers. And I hope PyLabRobot can be one of those tools. And then developers will take this and build like tools for the actual end users. So probably like the automation engineers, they take these APIs and they build like nice GUIs and nice interfaces for the actual scientists who are the end users of all of this. And so specifically, what are we working on? We're always working on adding new machines. You can see on our website the current machines that we support, and also the ones that are a work in progress. We're also looking to expand the documentation and the resource model. For example, we soon want to add nested tip racks and also start the protocol library. So we're looking at just open sourcing some of the protocols, because again, for the protocols, it's mostly people doing the same thing. So we would rather collaborate on that rather than reproducing the work over and over again for certain protocols. So that's the end of my talk. Thank you again to the supporters and to the users and the contributors. And here are some links. Thank you.
Thank you, Rick. Really nice talk, great overview. We will start with the questions now, and I already saw. We have one question from Basquin. I'm not pronouncing your name correctly, but are you unmuted still? still muted. Or if you like, you can also type it quickly in the chat and we can read it out if it's harder for you to. Okay.
Okay, so could you hear me now?
Yeah.
Yeah.
Yeah, sorry, it's the first time I'm using a Google tool.
Yeah.
Yeah, so I'm Jean Basquin. I'm basically sitting at the Max Planck Institute for Biochemistry in Martinsried. And so first of all, thanks for the great talk. And I think it's a great initiative that you guys are putting in place. So just